Hi guys, and welcome to another edition of Pink Biology. Happy New Year everyone. I hope this year is a better year and brings a lot of health and prosperity to everyone across the world. And this is the video to do with my university, and I went to Bournemouth. So I have mentioned their name here. They're not endorsing me, but I thought only fair as I did do the degree at the university. So basically we're going to see what I did in every year and a little bit of information to go with it, a little bit of science. So hope you enjoy. Okay, so first year we have got some bionomical classification. And this means that anything that is living has been named and discovered, hence why sometimes you have the year after things. So we've got an example here, which is human, which is Homo sapien, and that is Latin for wise man. Yes, we are. So basically here we have sort of an order of a group. So the classification would start very general. So you're either three main things. You're either archaea, bacteria, or eukarya. And basically what happens is that has to do with what type of cells you have. So for example, cells in plants, cells in humans, cells in bacteria are completely different, and they have possible things that are similar but are put into separate categories. So bacteria has its own complete category, and then it works its way down. So you've got kingdom, phylum, class, order, family, genus, and species. So species would be the largest group of organisms. So two of them would produce offspring, and that would include karyotyping and DNA, which we will get onto later. So let's just take it down and simplify a bit. So here we've got a flower, and I just picked it and it's completely wilted, but there you go, that is real life for you. So basically you've got an example of a genus of a bluebell would be a Hyacinthioides non-scripta, which is a bit of a mouthful. And this is why there's a lot of Latin. They were discovered such a long time ago. Hopefully when people find new things, they will be easy to understand. So basically what it would is it break down into what kind of family it would be in, and apart from what it's got sort of in the flower. So basically what its characteristics would be is construction, its heritage, what flower is it similar to. So that's how they sort of divide it up and then create names for different things. And we study quite a lot of plants. Now, forensic biology doesn't really think, in a degree you don't really think, oh, why are we studying flowers? But these are really key actually, because interestingly enough, if you find an unfortunate dead body in a crime scene, then the way that the plants around it have developed or grown or not grown or seeded actually can give you a lot of clues about how that person died and where that person died. So it may seem a bit trivial, but these actually are really, really good at identifying things in a crime scene. Okay, so here we've got an example of eukaryotic cells. Plant cells is one, and then we've also got animal human cells, and basically they come under eukaryotic. I don't get it right every time. I don't know everything, so I apologize if I might have got a mistake in there. But basically, they do have similar characteristics because they both need water, they both have nutrients and glucose and things like that to help grow and sustain. But they are very different. So some differences would be the walls are different, the cell walls. So you would have nutrients going in and nutrients going out. You have X phylum, which I remember is the X factor. So the X factor, if you remember One Direction, please endorse me. Basically the nutrients, they all go one way. That's how I remember that one. And the phylum is the opposites, they go all different ways. Basically in the cells, in a human cells, you've got, goodness knows, you've got so many things going on. You've got centrosome, microfilament, ribosome, mitochondrion. Mitochondrion is what you call the factory of the cell, so it'd be sort of maintaining, sorting all the things out, producing things, making things, nutrients. Then you've got the cell wall, which is predominantly got sort of a fat part and a water part. And what happens is the way that they join together, is they're semi-permeable, which means nutrients can go in certain ways and some nutrients and things can come out as well. When you've got the ions and calcium and goodness knows what, there's loads and loads and loads of things. I find cells really interesting because we are all made up of cells, and it's amazing to think, like a hand, for example, there it is, a hand is made up of many cells, and just think how you started as an embryo in the womb. You started from 2 cells and then you just create this human. I mean, it's just absolutely amazing, so much to learn. It's always good to know the basics though, because it can get extremely complicated. We're going to have a look at what a lysosome does, which is in a human cell. Okay, so I actually learnt about cell fractionation in university in level 1. You can see the top left hand corner. Lysosomes specifically, what are they? Who knows? They actually, and I've got some notes here because I can't remember it all, they actually are organelles covered by a membrane cell, and they contain digestive enzymes which break down complex molecules and sugar, protein, etc. And these enzyme catalysts can be found in the mouth and stomach, and they basically create apoptosis. Now, medical terminology, we haven't learned that yet in one of my videos, but we will. So apoptosis is a programmed cell death. So it means that the cells that aren't working anymore are killed off if they're not useful anymore. It's also called phagocytosis. So phago is when you're eating or engulfing, if you like. So phago, so let's use a flower as an example. So phagocytosis, instead of way of killing a cell, you would have the mechanism actually come and just sort of like eat it and then absorb it. There we go, dead flower. And that's another way of how the body gets rid of cells that it doesn't need anymore, because a cell rejuvenates, your tissue rejuvenates. If you cut your hand, I'm using the hand again, the cell comes away, the cells come away, they sort of clot, and you've got the proteins and you've got all the things that the clotting factors, and then the dead tissue comes off, and then you've got rejuvenated skin. So how does that work? Your body creates new cells, but obviously you've got dead cells in. So if you have dead cells inside your body that are no longer useful, this is what lysosomes did. And basically, if you look at the picture on the right hand side, you can see real microscopic images of lysosomes, and to me they look a bit like cabbage. But basically it was a really interesting piece of coursework, and I did this for first year, and it was really tricky because it was something that I didn't really understand, but now I do. So they're really, really interesting. And just importantly, just to point out that the microscopic lens was times 400, which is really crucial because there's different types of lenses that can see different types of things during a microscope. And I also learned how to use a microscope, so there you go. Okay, just a few more words about lysosomes again. So basically we're just looking at some sort of scientific knowledge of words just to understand. So on the right hand picture at the bottom it says lipid layer. So this is when I was explaining you've got the fatty layer, and what happens is that you've got hydrophilic and hydrophobic, and hydrophobic means afraid of water and hydrophilic means love water. So basically it means opposites attract in a way, that the way that the cell wall is set down, basically that's how it lets nutrients in and nutrients out. Then you've got membrane transport proteins. So these are the proteins that will help take a cell substance and take it in and out. So for example, if you've got a cell, say we've got a sellotape here, and this is a substance that's going through a cell wall, and what it would do, it was sort of attached to the protein, and then it can pass through the cell wall, and it couldn't pass on its own, but through this attachment it can pass. And then you've got, I'm not sure if it's in lysosomes, but you've got the membrane gates which sort of open and shut, sort of let the membrane in like that. A bit like a turnstile that we have in England where we keep cattle in from getting outside of a farm. It's sort of you go in one way and then you have to shut the door behind, then you can carry on. And so basically there's just little bits and pieces here. Hydrolytic enzyme mixture, plasma membrane. So plasma, you might remember, is something that's in the blood as well. So similar constitute. And then you've got all different bits and pieces on the other side. You've got Golgi apparatus, cytoplasm, vacuole, rough endoplasmic reticulum, nucleus. So nucleus is where the DNA is. Important, important, important. And we're talking about that a bit later. So we're just nearing to the end of year 1, and we're going to look at protozoa now, which are single-celled eukaryotes, and they are either free-living or parasitic. So parasitic means they can't live without a host body. So for example, that could be a virus in the body. It needs cells to attach to. It looks a bit like a robot, a bit like this, with like little legs, and the legs actually attach and embed into the cell. And basically, this page is about just looking at the different binomial nomenclature again. So the naming process, the posh word for the naming process. And this one is a eukarya. The kingdom is Protozoa. And as you can see, its species is Parasmecium Aurelia Baceria. Don't know if I said that right, but that's a lot to say actually. So basically, we're just looking at these under the microscope in the labs. We did a lot of laboratory work. It was really, really interesting. You wear the white coat, very strict instructions how to do everything. But yeah, it was really fun. So it's just a little bit about that really. And you can see there's little cilia. So cilia is very similar. Cilia is like this. And it's very similar to what you have in a bowel, where actually the cilia helps move the food go down the lining and actually absorbed into the bloodstream. So yeah, okay. Okay, we're on to year 2 now, and this is about genetics and DNA extraction. Biochemistry, yes, I was completely lost, and yes, I wrote a gallon of notes. Here you can see in my lab book on the left, I wrote everything out because I didn't understand it at all, and so I just had to learn and learn and learn and learn and learn. So DNA extraction is used for genetic causes of disease, drugs, diagnostics, forensic science, sequencing, goodness knows, there's a whole load of things it's used for. And basically, so DNA is the genetic makeup of what makes you you, I guess. So what makes you have the characteristics, what medically you can cope with, what medically you can't, what illnesses are in your family. Genetics and DNA are basically the building blocks. They are who you are, and they dictate what cells do, what cells don't do. So they're really the brains behind the human body, particularly, which is absolutely extraordinary. And here you can see an example where you put, see if you've got a DNA example. So you could say there's some DNA at a crime scene, and obviously you can't pick up pure DNA. There's no such thing. It's messed up with everything that's on it. It could be on the ground, could be mixed with other people's DNA. So the sample will be put into like a little test tube, and then it would be put into a centrifuge, which is the machine that goes really, 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 really fast, okay? And it spins, and by the ways it's spinning, it's actually washing and drying and separating the different parts of the DNA. So, and sometimes it's not always very clear, but there are ways of learning about it and understanding of alleles and, oh my goodness, so much. It is such a complicated thing, genetics, but really, really, really very interesting. Okay, carrying on to year 2. Now, I haven't covered everything that I've done at university because I did so many different units. And if you saw on the first page, it's just forever. It was quite a new degree, forensic biology, but it's a lot of science, as you can imagine. It wasn't just science, it was law, it was chemistry, biochemistry, cells, crime scene. Goodness, it just went on and on and on. There's just so much I covered. So this is just a snapshot, really. So basically, this is about preparation analysis of aspirin. Now we're going sort of down the medical route, which really interested me. Medicines and toxins are a big passion of mine. I find them really complex and sort of like puzzles, piecing things together and how they work. Now, as you can see, the chemical symbols absolutely terrified me. I didn't know what they were. I had to learn all of them, and I didn't even know what double lines mean on the pento type of the sort of what I call octagon. It's not an octagon, the octagon's 8 sides, so that's 1, 2, 3, 4. So that's 6 sides, so hexagon. I didn't know what the lines meant. I didn't know what the symbols meant. CH3, who knows? I got completely confused. So I had to learn basically from scratch to understand everything to even get to this point. So basically, this identifies organic, polymeric, and inorganic materials. So a Fourier transform infrared spectroscopy, or an FTIR analysis, is where it is looking for, so poly is sort of plastic, poly is polymeric is plastic, so poly is many, inorganic, which means it's not organic material, so things that could be found. So for example, you could say a piece of paint. It says like a paint scratch on a car, a piece of car paint body was found at a crime scene, then they could identify where that came from, what make it was, things like that. But in this case, we used it for analysis of aspirin. So we actually made aspirin, which is a painkiller in the UK. Pretty cool, pretty cool. We did actually make like a white powder. It was actually really interesting. So there's all the technical ways of how it's analysed, but it was really, really good, and I learned a lot in the labs again. Okay, so I'm going to refer to my notes again because I can't remember all this, to be honest. Basically, this is the preparation analysis of aspirin. So as you can see on the bottom right of the lab book, it's where my aspirin was made, or crude product. Now, crude product means it's a very original state, so before it's had different chemicals and processed into the actual little pill sample that it looks like. And basically, Fourier transform infrared spectroscopy is electromagnetic radiation with wavelengths longer than visible light, and it's the absorption of emission of a solid, liquid, or gas. So basically, they put light into a substance, and what it emits back to the machine is what it clarifies what it is or it isn't. So for example, you might have a piece of car paint from a crime scene, as I said before, and then they put it in, and then they can analyze and work out what materials it is, and then match up to see if it does actually resemble that. It is a key piece of instrument. It can detect drugs. It possibly might be used on border controls and things to detect drugs. I'm not sure. Possibly it could be. Look it up. It'd be quite interesting. Yeah. So basically, this is what people think of as a DNA, and this would be described as a DNA ladder. It's a very basic DNA at its very simplest form, and the way it works is that it's folded like this, and that's how it fits. And we have many of them in our body within chromosomes, which are the pairs that make us us. So basically, a way of using fluorescence in situ hybridization would be to identify if someone, for example, in a pregnancy if the baby has Down syndrome. And Down syndrome is looked upon as a genetic deformity, but I see it as someone's individuality, if you like. People always bring what they do to life, no matter who you are. And so basically what you would be looking for is if they had an extra chromosome. An extra chromosome would be called trisomy. Now, tri means 3. And so basically it would mean instead of having 46, they would have 47. And lots of genetic illnesses and things can be identified. So Huntington's disease, they can look for cancer, lots of different things. And obviously the general, if you are a girl or a boy, or if you're maybe a mix, who knows. So you've got the X and the Y. So the XX means female, XY means boy. And if you look again at the top right picture, at the very bottom right, you can see an X and a Y. And it is the male that determines whether the child is a boy or a girl. So basically, I'm going to do a video on genetics to explain this in more detail. It is very complicated, and you have to understand the very basics to get to the point to actually realize why you need ideograms, which is the top image on the picture with the yellow bands, and why you need to learn about the chromosomes and DNA and what they can actually do. DNA can dictate whether you are susceptible to paracetamol or not. There are mutations, insertions, deletions. It's very complicated, but it's really amazing and it's worth having a look at. Okay, next slide. Now, because we're on year 3, it gets complicated, and I did a lot of studying this year, also doing my dissertation, which I'll talk about in another video. And basically what we have here is plasmid DNA purification. And my goodness, was this complicated. So basically you've got plasmid DNA. So the DNA would be in circles, and what it would have is identifications of different types of DNA within the circle itself, so the plasmid. So for example, I've got a makeshift plasmid here with foam, and what it would look is you would take out certain pieces out of the different parts. If you can see, you've got different colours. Hopefully that's coming up there. You've got, it's actually pink, but it's not coming up. You've got pink, purple and blue. And what it would do, it would have different sections, and it would actually cut pieces out. And then you could insert pieces in. And so basically you're looking at where it says GCTTA, that's actually the pairs of the DNA. That is the basic names of these pairs. So you would have AT and CG. And although it's very, very complicated, it was really, really interesting. But this was high biomolecular stuff. And yeah, it was very confusing, but it was so fascinating to how you can actually manipulate and change DNA under a microscopy, so minute, you can even see with your eyes. Moving on. So as you can see, we've come to the end of that whistle-stop tour of my degree at university. As you can see, it's very complex and very, very confusing, but it was so interesting and I covered so many units. Shout out to Forensic Corner on YouTube, who have supported me and I have worked with in some videos, so thank you very much. And here you can see my details. We've got LinkedIn, we've got Pink Biology email, we've got my Twitter, we've got Pinterest. Like and share my videos. I do this for fun. I do this to educate and to make science a bit less scary. Albeit this video wasn't about that really, but it's just about introducing things and letting people understand about science and making people involved. So thank you very much for watching, and I'll see you next time.